Bacterial cell wall is made up of N-acetyl muramic acid and N-acetyl glucosamine held together by peptide chain. It gives rigidity, density, and protects it from environment. It is described as a tire outside the tube. Tube is similar to that of cytoplasmic membrane. Uh, it gives shape depending on the shape of the bacteria. Bacteria are classified into cocci, which are spherical in shape, rod-shaped bacilli, comma-shaped or vibrio, spiral flexible or spiral rigid or spiralum, spiral flexible or spirochetes, and hair branching bacteria. There is cell wall-less organism also called, which is Mycoplasma pneumonia, which gives rise to pneumonia. Cell wall is one of the important structures of bacteria, and it cannot be seen. You have to do micro dissection, and you have to do the special staining to see the cell wall. Cell wall of gram positive is more thicker than gram negative. Gram negative cell wall is thinner and it is more complex. It's made up of more lipid, and gram positive cell wall uh, is contains two additional chemicals called magnesium ribonuclease and teichoic acid. And gram staining, because gram negative has more lipids, it gives away the diadine complex, which is formed after putting mordant, alcohol being alcohol or acetone as a decolorizing agent. Cell wall, when it put in a hypertonic solution, uh, all the cytoplasm goes away. It's called as plasmoptosis. When it put on hypotonic saline, it swells up, it shrinks, it's plasmolysis. When it's grown in presence of penicillin, cell wall loses cell wall, it becomes pyroplast and protoplast. They are spherical in shape with respect to gram positive bacteria. There are special forms which are called as L forms when you grow the organism in presence of cell wall inhibiting substance. They are called L forms because they are discovered in Lister Institute of London. And these L forms, when again they are grown in enriched media, they revert back to the original forms. So important part parcel of things you should remember about cell wall is it is a made of N-acetyl muramic acid and N-acetyl glucosamine held together by peptide chain, and it is important for maintaining the shape of the bacteria. Cytoplasmic membrane is considered as lungs of bacteria and helps in the division of the microorganism. Cytoplasm is a colloidal material which contains inorganic and non-organic substances. It contains nucleic acid, nucleus, and inclusion bodies and other things. Bacterial nucleus, important function of bacterial nucleus, it important structure which contains characters to form progeny, and uh, it is made up of single molecule of double stranded DNA. And there are granular substances which are called RNA. They are synthesized protein for bacterial cell. When they are tagged with, they are also called messenger RNA. They help in the translation of bacterial proteins. There are special inclusion bodies which can be made up of carbohydrate, proteins, and fat. There is a special inclusion body which helps in the identification of bacteria. These are called metachromatic granules, pop babes, or moletin granules. There is a special structure called plasmid. That's extracellular genetic material present in bacterial cell, which transfers drug resistance and toxigenicity. This is all you should remember about bacterial cell wall, morphology of bacterial cell. You have to write a neat diagram of bacterial cell, and sometimes full question will be asked on the bacterial cell morphology. Thank you very much.